Let's just imagine for a second that each one of your cells has an appetite. Perhaps one cell in particular has a chocolate addiction. Can you imagine what might happen if this cell saw a plate of brownies? What about a chocolate milkshake? Could a cell figure out a way to transport that across its cell membrane? Where there's a will, there's a way. If your cell is as much of a chocoholic as we're describing, it might be able to figure something out even though it doesn't have a mouth like you or me. We've talked about active and passive transport mechanisms used to move substances across the cell membrane. In this lesson, we'll focus on additional methods of transportation that allow some cells to eat or drink its surroundings. In addition, we'll discuss how a cell digests these items and what happens to the leftover food that can't be digested at all. In order to be able to chow down on substances in its environment, a cell will actually engulf its surroundings, folding its cell membrane around whatever it wants to ingest as if it's taking a giant bite. This process is called endocytosis, or the import of molecules and particles into the cell by folding the cell membrane inwards. The prefix endo means within. During endocytosis, vesicles or circular buds comprised of membrane are formed around the substance to be digested. These vesicles are then transported into the cytoplasm. There are 4 types of endocytosis, 3 of which are based on clathrin and 1 that is not. Clathrin is a component of the protein that coats vesicles as they form from the plasma membrane. The first of the 3 clathrin-based types of endocytosis is phagocytosis. Phagocytosis comes from the Greek word for devour. Phagocytosis is the process where large molecules or other cells are eaten by engulfing them. In phagocytosis, the cell membrane folds around the object to be eaten, creating a vesicle that can then be internalized. For example, a white blood cell in your immune system can eat a foreign bacterium whole in one bite. The second type of clathrin-based endocytosis is penocytosis, which comes from the Greek word for drink. It is the process where small dissolved substances and liquids are taken up by a cell. Penocytosis is similar to phagocytosis. Both of these processes allow cells to non-specifically take up substances from the surrounding by engulfing it with its membrane. However, in penocytosis, smaller vesicles are taking up liquid and dissolve solutes in that liquid, such as these small squares here, rather than large molecules. For example, the cells surrounding your blood capillaries perform penocytosis to drink your blood, while at the same time taking up any solutes dissolved in your blood. In the third type of clathrin-based endocytosis, the cell membrane also folds around a substrate to engulf and internalize it. However, in receptor-mediated endocytosis, this is a process that recognizes and targets a specific substrate through the help of integral membrane proteins on the cell membrane surface. Here, integral membrane proteins called receptor proteins recognize and bind to a specific target, such as these stars. In this example, these receptor proteins would not recognize and bind to non-star objects, like the squares. When a receptor protein binds to a target, it signals the cell membrane to enclose the target and internalize it inside a specialized vesicle. Phagocytosis and penocytosis are non-specific methods of endocytosis. They would eat any kind of chocolate. Receptor-mediated endocytosis is specific, picky. It knows exactly what it wants. The term caveoli is Latin for little caves. Caveoli are pockets in the plasma membrane, which were first identified with an electron microscope in 1953. Caveoli do not have a clathrin-based protein lining them. Instead, they have caveolins, which are membrane proteins that play a role in various metabolic pathways and cellular signaling. In mammals, there are 3 types of caveolins, which are, you guessed it, caveolin 1, caveolin 2, and caveolin 3. Cardiac and skeletal muscle cells employ only the caveolin 3 type of protein, while smooth muscle cells employ all 3 types. Caveolae-based endocytosis